Hello, I'm Dr. Hagop Kantarjian from the Leukemia Department at MD Anderson. And today I have the honor and pleasure to interview Dr. Michael Mauro, who is the leader of the CML program at Memorial Sloan Kettering. He is also one of the great innovators in CML research and in advancing the prognosis and outcome of patients with chronic myeloid leukemia. So, Michael, today I was fortunate to attend your talk on ELVN-001 in chronic myeloid leukemia. So I'd like you to introduce to us what this molecule is and what it means in CML research.
Oh, first off, thank you for that very kind introduction, and I'm glad to be here to present this data. So ELVN-001 or ELB-001 is a novel ATP competitive or active site inhibitor for chronic phase CML, or at least for pH-positive leukemia. It's being tested in chronic phase CML. The idea here is we've been looking for more potent or best ATP inhibitors for CML or pH-positive leukemias in general, and we've had many approved and we have many in trials. Some of the goals are to develop something that's more specific, better tolerated, narrower spectrum of toxicities. So preclinically, ELVN has very specific and potent ability to bind the ABL wild type, as well as cover mutations with sort of an even ability to cover common mutations like T215I and active site mutations, as well as myristoyl pocket mutations. And additionally, it seems to not be likely to be a substrate for efflux pumps or competing metabolic pathways such as the CYP3A4 system in the liver. Lastly, it doesn't require any food exclusions, so it has a very good preclinical profile.
So let me ask you, for people who are not familiar with the BCR, ABL, tyrosine kinase inhibitors, so we know that imatinib is the first generation TKI. Then we had the second generation TKIs, which were more potent, dasatinib, bosutinib, and nilotinib. And now we're talking about the third generation TKIs, which are not only more potent, but are active against T315I. So how does ELVN-001 fit into that spectrum of the third generation TKI? How do you classify them?
That's a great point. The field evolved just as you stated, and I think with this next generation or these newer TKIs, we've been looking for, again, best-in-class ATP competitive inhibitors. Ponatinib was, I think, the next, or the next drug beyond the earlier drugs to be developed, which was a huge advance, a much more specific, potent ABL inhibitor, again, ATP-focused, that was able to overcome T215I. Olverambanib is a drug that's been developed and approved ex-US, now studied in the US, led by your center and others. And the hope is, I think, that we can have perhaps multiple ATP competitive and maybe better tolerated versions in that class, as well as allosteric or specifically targeting the myristoyl pocket inhibitors or STAMP inhibitors, which some people would also say fit into the third generation or the next generation TKIs. We have asciminib approved there and many drugs in development, TERN-701, and Centage, and so on. So I think we're seeing really still a lot of activity and research here.
So let's talk about ELVN-001. Tell me a little bit about the study design and what kind of patients you accrued to this study.
Sure. So classic phase 1 design, dose escalation, 3x3. Probably important to point out, when a trial like this is launched in the current year, now we're dealing with a somewhat different population of patients. You're going to have patients with multi-TKI resistance and intolerance, generally 3, 4, 5 TKIs for the majority of patients. They may have had resistance or intolerance or both. I think the majority here were resistant to their prior line of therapy. And you do have now asciminib and ponatinib exposed patients, which is a really important question. As good as those drugs are, we do have both intolerance and resistance. So that's the playing field. Phase 1A was completed, and then naturally you optimize the doses, and a target dose was chosen in phase 1B, as well as a randomized sort of Project Optimus type approach per the FDA to move the drug forward. Those cohorts have all essentially accrued at this point. We're the last patient to accrue into the phase 1B.
And what is the final dose that was agreed upon for the phase 2 study?
I don't know if we have that answer yet. I wasn't privy to those conversations. I expect it may be 80 milligrams daily.
And tell us a little bit about the results. So, did you see better responses at the higher dosages? What kind of overall complete cytogenetic responses and major molecular responses did you have, and how durable were they?
So, probably first, should look at the safety. Of course, those are the real goals is to understand the activity and the signal. But I think one of the stronger points coming from the study so far is the safety. So again, a heavily pretreated population, many with prior TKIs with potential adverse effects, and a bit, an age where comorbidities are pre-existent. We had no dose-limiting toxicity. We had very limited grade 3/4 toxicity, probably what you might expect, some myelosuppression, and rarely leading to modifications. With regards to the patients that couldn't tolerate the drug, there were some adverse effects which may not have been related. There's about to study discontinuation, thrombocytopenia in 2 patients. So, and then lastly, much like the other TKIs, the toxicity may not be dose dependent. It didn't seem to change as we went through a full range between 20 and up to 120 milligrams.
So very safe drug so far with the phase 1 experience. How about the efficacy?
Sure, I didn't answer your question. So the efficacy signal looks good. Now, a study like this is going to have patients entering in cytogenetic or molecular response. So nice to say that in CCYR and MMR, cytogenetic and molecular remission patients, those were all sustained, which is important because, moving someone to a therapy they're able to sustain a safe response in is important. With regards to gaining, cytogenetic remissions were gained in the majority of patients who were lacking. I think it was roughly 50%.
I saw about 50%.
Yeah, which is actually... And we have to, pick our battles. And a multi-drug resistant CML, a stable cytogenetic response is meaningful. Major molecular responses in, I think, a minority in the 30 to 40% range. The questions we now have to focus on is, a more uniform population and maybe some of the specific questions. T315I, some of the points you raised in your question, durability over time. We do have a patient treated, I think, more than 2 years. We have a good number of patients more than 6 months. So I think it does look quite promising from the efficacy signal.
And the future plans for this drug?
Great question. So the field, as you said, is broad, but I think we saw a nice advance with ponatinib, a huge advance, to be able to treat T315I and resistant patients in advanced disease, as you've studied extensively. So in the breadth of pH-positive leukemias, if we had other ATP-competitive drugs that were very selective, that were safe, that we could use alone in combination with the allosteric inhibitors, for example, I think this is an exciting area to move into. But right now, the next steps would be a proper phase 2 trial with the chosen dose, probably saying this could be then a preferred ATP-competitive inhibitor, perhaps as a second line agent after a previous failure, a failure after asciminib, and then the future, beyond that remains to be seen.
And any plans for Philadelphia positive ALL, since we know that the TKIs with blinatumomab are doing so well? Any plans of developing ELVN-001 in Philadelphia positive ALL?
I think that's a great question I should bring back to the folks at Enliven. I don't know if they've developed trials in that area yet, but I think the non-chemotherapy or TKI and low risk novel agents in ALL and lymphoid blast crisis is looking so promising. So I think that'd be great to explore.
Thank you very much, Dr. Mauro, and I wish you a lot of luck with the continued research with this very exciting drug.
Thank you.